So good morning, everybody. My name is Ludovic Besaury. I'm an associate professor in the same lab as Gwenel, and I'm working more on the biotechnology part regarding the valorization of lignocellulose, but I have worked in the past with microbial communities from the environment. That's why my topic is like this today. What this slide has already been presented before. So as you know, we have a lot of the estimated world population is increasing, and we have less and less, and we need to decrease our consumption of carbon fossil compounds. So an alternative has been to use lignocellulose, which are compounds that can be integrated into the bioeconomy, and for that bioeconomy, this lignocellulose can support for energy, biomolecules, and agromaterials also. And so in that biorefinery that we are working on in the lab, so as Gwenel already explained, it's a very complex and very recalcitrant material composed of several polymers. And it can be turned into different type of products that I have presented. And there are many recovery processes that can exist, such as physical, chemical, or mechanical. But those one are going to be not green enough or going to consume a lot of energy. So the goal is to use biological processes that occur into the environment and to study the degradation mechanism of those degraders, such as bacteria and fungi, and to apply them into the lab to produce some compounds of interest. So in our lab, what we do is that we produce enzymes, and we study enzymes communities, and we study the production of molecules using lignocellulose as starters. So for that, we do either enzymatic fractionation of microbial bioproduction, and we are going to use microorganisms of interest or environments of interest. We are going to use a different type of biomasses by taking into account their recalcitrance. So that means their amount of lignin, hemicellulose, cellulose, their structure. And we are going to use to study the microbial strategies and physiology using the same techniques as my previous colleague said, using multi-omics. And we are going to use some different strategies using bottom-up approaches by integrating microorganisms each after one, or by doing top-down, so by reducing microbial communities from the environments. To release some logs, and in my research program, there are two axes. Axis number one is to study and to identify new active microbial population and to characterize new efficient enzymes, because we need to provide some new enzymes to be able to deconstruct that lignocellulose efficiently, and then to perform bioconversion of lignocellulose into some products of interest by using this lignocellulose as a carbon source. And so we will try to produce some bioplastics, pigments, methane, and to turn lignocellulose into bioelectrochemical systems. So due to the topic, I'm going to talk now about the top-down approach. And in the top-down approach is to reduce the complexity of microbial community, and to reduce that and to have only key efficient players, because in the microbial community, you have some cheaters that are there that are going to use the work done by the other microorganisms. So for that, we use a specific environment, which is the phylosphere. So, and it's just the phylosphere of some residual AgroResources. And so we used that some with bran and some with straw. And this is just our inoculum with already the carbon source. And we studied the evolution of those phylospheric microbial communities that are already present onto those those compounds. And we studied that to study the evolution and the dynamic of the production of lignocellulosic enzymes to see if we were some new lignocellulosic strands able to deconstruct the lignocellulose with new enzymes, and to study by doing some metagenomics what can they do and are there new enzyme there. So the results are enough significant so that I don't have to present as beautiful figures that my colleague just before, but this is just what she presented. So when you have the phylosphere of wheat bran with WB and wheat straw WS after 48 hours, you have a shift in microbial community composition. And what was interesting that when you look at that, you have some new key players that are there, that are present, that were never presented in the literature, and that can help deconstruct the microbial community. So we isolate some of them, not all of them. And so we have now some efficient microbial communities able to deconstruct. And when we looked at when we reconstruct some mags, as my Finnish colleague presented before, we were able to reconstruct some mags, some metagenome assembled genomes, and in some of them we were able to identify some new enzymes called carbohydrate esterase for Acinetobacter, which were, for example, never identified into the Acinetobacter baumannii core genome. It was the same for the Pseudomonas WB bin04, which was able to encode a lot of auxiliary activity enzymes, some AA2, AA3, and other on that were never detected into some already sequenced Pseudomonas genomes. So that means that starting from a very stressful environment, which was the phylosphere, because there is not a lot of moisture, not a lot of available carbon, those microorganisms were able to develop some new enzymes and to have some new genes coding from some new enzymes that were not described already in those specific genera. So now, since we have the sequences, we can do some synthetic metagenomics, synthesize them, and study and characterize those enzymes by DNA. Another point that we did in our research in the lab was to do a bottom-up approach, because this is the other strategy, which is easier. And the bottom up, so it will be the another word is like called the co-cultivation, because you know that in nature it happens in the syntrophic process. There are a lot of publications dealing with the degradation of microorganisms, but only by studying one bacteria or one fungus. And the goal is to do some co-culture with 2 or plus microorganisms. And so this has been done a little in the past with organisms from the same species, like from E. coli or from the INRAE kingdom. And so, as you can see, the simplicity is related to the diversity, because when it's E. coli, it's the same media, but when it's for the INRAE kingdom, you have so you can imagine that you will have more competition, a different type of media that you have to take into account. And here we try to do that with inter-kingdom. And as you can see, it's coming to the literature. It's arriving in the literature because the number of paper dealing with that is increasing. So it's a new hot topic in terms of biotechnology. And there is a huge advantage into doing some co-cultivation, is that you will have a distribution of the different functions between the 2 organisms or 3 organisms that you will mix into your reactor. And so that will help to prevent any nutritional deficiency, because since you will have that crosstalk between the microorganisms, you will always have some exchange of metabolites there. And so for that, we isolated the Aspergillus niger strain, we sequenced it, and when we looked at his content into the caseins which is related to carbohydrate active enzymes. So it has a lot compared to the already known Aspergillus niger strain that are in the literature. But what was more interesting that when we did the co-culture, we did the same annotation with the 3 strands that we were going to use. 3 bacterial strands that are Streptomyces, Avermithylist, Coelicola, and Glyceremens. And when we looked at the Venn diagram of the different functions, the different families that represent those auxiliary activity, those carbohydrate esterase, glycoside hydrolase, polysaccharide layers, so those enzymes that are involved into the lignocellulose deconstruction. Well, the Venn diagram, they show that we have, of course, a common core of the same function, the same families, but there is also each microorganism has its own specific amount of casein. So that's led to a greater diversity of casein families into the coach culture, which we hope will allow a better fractionation of the biomass. And moreover, we had a specific arrangement in glycoside hydrolase, which are the key enzymes for the lignocellulose deconstruction. And when we mix the different microorganisms together, and we followed that through time, we saw where, for example, at the beginning at 48 hours and after at 140 after 7 days, well, we had the main the fungi that was able to deconstruct the biomass by producing xylanase, but at the end, when it was mixed with certain type of Streptomyces bacteria, the coelicolor here, for example, here and here and here and here, it was able to, we had a greater production of enzymes, especially for the xylanase and some debranching activities, such as the glucosidase or the xylosidase here. So, which is interesting is that when you have a certain mix of fungi and bacteria, you have an elicitation. So that means that you don't have an organism A and an organism B, and it doesn't make A plus B, but it can make A multiplied by B because we have an elicitation up to 8 times. So that means that we have an enhancement of the biomass deconstruction. The other interesting part with that is interesting result is that the carbon source, and lignocellulose is going to act as a compound which will help for the elicitation of silent biosynthetic clusters. So we did some metabolomics here, and we did those co-culture with glucose or with wet bran. And we did that either with 1 or 2 microorganisms. And when you have the PCA that is here, well, you can see that when you have some a mix of when you have the mix of the microorganisms together, you have a different pattern regarding the metabolite production. So that means that there is always a lock for the expression of those secondary metabolites. One possibility was to use different type of carbon source with what we did, but the other type was to make a cultivation that helped in the past by producing some new metabolites. But another possibility is to use some lignocellulose, which is not always done in the literature. And because that's new, because it allows to highlight those biosynthetic cluster genes, because those microorganisms, they are more used to see those carbon source in the environment, so they are happy to see them back in the lab, in the flask, compared to some other minimal media that we can give to them. And the other thing that we did regarding that approach with co-culture is that we were able to do some dual RNA-seq. So that means that in the same flask, we were able to quantify and to study the transcriptomics of the of only the bacteria and after only the fungus. And we did that with Trichoderma and the strain Streptomyces violaceo. And I'm just showing you the results for the bacterial strain. But here, as I said regarding the biosynthesis of secondary metabolites, it has been enriched by the communication that occurred between the fungus and the bacterium. So you have KO genes that were more expressed by that condition. And also that communication between the two microorganisms help them to express more genes regarding the lignocellulose degradation, such as amylase or polygalacturonase. So that's exactly the kind of result that we wanted, to have co-culture that communicates and express more genes for the lignocellulose degradation. For the last part, quickly regarding the bioplastic bioplastic production. So, for example, as you may know, we looked the main one found regarding the industrial production is the polyhydroxybutyrate. So it's a bioplastic that has a lot of properties of interest, and it's a linear polymer here that can be produced under stress condition and used as and it is a storage compound. So we looked at that because that PHB is produced by bacterial synthesis, but we are going to for that we use, or the industry use biomass hydrolysates. But those biomass hydrolysates, they are going to use a lot of pretreatments, which I explained before, are not green or are going to cost a lot. Or either you are going to use some sugars or glycerol already available, but this is going to cost a lot of money also. So that means at the end you have increase in production cost, so a decline in competitiveness and a price which is going to be higher compared to polypropylene. So a solution was to use the lignocellulosic biomass as a substrate. And as you can see, the cost of biomass is lower compared to a cost of glucose per ton. But in the literature, there is no microorganism able to use direct or raw lignocellulose and to turn that into bioplastic, into PHB. So we were looking for some communities able to do that in one step directly. And so we did that with a forest soil. So we used the diversity of forest soil by doing some sequencing. And when we add some wheat straw, we saw a clear decrease in diversity of the community, which was dominated by this blue bar, and this blue bar corresponding to the Parabucholdaria. And Parabucholdaria is a non-genus able to produce some PHB. So here we have a microbial community here that is able to deconstruct wheat straw, recalcitrant biomass, and has a huge potential in PHB production by using raw lignocellulose. So for that, we did some isolation of the strain, different strain that we had, and those bioplastic PHB are produced by different type of enzymes, such as by different type of genes, such as PHA, A, B, and C. And we have a huge number, and we have more genes coding for those enzymes, for example, for this strain that dominated the microbial community. We did some fermentation tests using lignocellulose as carbon source without any pretreatment, and we were able to produce using that within this bacteria a lot of bioplastic without any pretreatment, but it remains lower compared to the use of pretreated biomass. So the possibility in the future would be to use those microbial communities, not one bacteria, but those microbial communities, and to make a production using mixed microbial communities to produce that this PHA or this PHB at the end. To finish, I would like to thank everybody that participated to this research, and I would like to thank the organizers for inviting me.
You're muted, Hélène. Sorry, I'm muted. Thank you, Ludovic, en tout cas, for this presentation and to see the nice opportunities for Parabulgoldaria. That's quite interesting to see what can be done with that bacteria. So, is there any questions? Maybe not yet. So maybe I'm just going to start the first question. Maybe other questions will come. I was wondering, I mean, how can you analyze the capacity to synthesize that kind of biofilm or bioplastic, as you said? I mean, how do you do that in the laboratory? How does it work?
So for that, we have the first we run the microorganism into a bioreactor. We cultivate into a bioreactor. Then we have the bacterial pellets, and we are going to extract using solvents, so which is not green for this step at least. So using mainly chloroform, and then you can dry the lysate, and you can filtrate and dry the lysate that you have, and then you can study, you can weigh, and after measure by doing some mass spectrometry or FTER, the compound polymer that you obtain.
And how long does it take to synthesize? I mean, to develop those kind of films?
The inside the cells
process?
is going to take no more than 24 hours.
Yeah.
And after to obtain the final product with the steps that I just described, it's going to take 3 days, no more.
Yeah.
Just to dry.
Like fungal growth. Yeah.
Yeah.
Okay.